So this is the recorded lecture for Chapter 2. The title of the chapter in this textbook is Chemistry Comes to Life. And one of the really common questions that we often have when we start to, here we go. One of the really common questions that we have when we start to do a chapter on biochemistry in a biology class is, why are we studying chemistry in this biology class? And it's because at the root of all biological organisms and functions, there are molecules and chemical reactions that are taking place. So although we are living beings, at the root of us, we are chemicals and molecules that are functioning and interacting. So we kind of have to understand some of the fundamental concepts of chemistry as we start to go through and look at biology concepts and processes. A lot of what we're going to talk about in Bio 130 is going to be based on your understanding of this chemical basis of biological systems. And there's a number of other chapters that we will go through where we will refer back to Chapter 2 and Chapter 3 on cells to remind ourselves about some of these basic building blocks of living organisms as we talk about what those molecules and cells do in the digestive system and the respiratory system and the cardiovascular system and the skeletal system, for example. So we're starting with the basics of chemistry. Chemistry is not a prerequisite for this course. I'm not making any assumptions that anyone has chemistry in their background. So we're going to start really basic. We're going to start by talking about the structure of the atom. And so when we talk about the structure of the atom, there's two parts that we're going to talk about. We're going to talk about the nucleus, which is the part in the center. And it has positively charged particles called protons and some neutrally charged particles called neutrons. And then circulating around in electron shells or energy spaces or energy levels are negatively charged particles called electrons. So a lot of times we denote protons either with a plus or P plus. We denote electrons often E with a negative charge. And then neutrons don't have a charge. They're neutral. They're not positive, they're not negative, they're very neutrally charged. And so this is a figure from a different textbook that kind of helps us break down this in structure. This is similar to a figure that's in your book. And so the nucleus is the area on the inside, and it's comprised of the protons and the neutrons. And then the electrons circulate around the nucleus. It's really more similar to this electron cloud model. They're found circulating around the nucleus at different energy levels. In biology, we stick to this electron shell model, sometimes called the Bohr model of the atom, B-O-H-R, named for an old white guy scientist. Because what we're really concerned about in biology is not these atoms individually. We are interested in how they form molecules and how they go through chemical reactions. That's what we want to understand about them. And the way they're going to form molecules and the way they're going to go through chemical reactions depends on this outer energy level, or this outer electron shell. So we're going to use this shell model to help us understand what this atom is going to do. Because by the end of this chapter, we're going to start talking about bigger molecules, proteins, carbohydrates, lipids, nucleic acids, and that's what some of your assignments and discussion boards are going to be looking at. But we have to kind of understand some things about this basic structure before we get to those bigger molecules. So this is a figure that shows all of these different structures of the atom for all of these different atoms. You don't have to memorize any of these structures. One of the things that you'll notice in atoms, atoms are electrically neutral. So their number of protons is going to equal their number of electrons. The charges are going to balance out. The number of positive charges is going to equal the number of negative charges. So overall, an atom is electrically neutral. So carbon has 6 protons, and it's also going to have 1, 2, 3, 4, 5, 6 electrons. Same with nitrogen, 7 protons, 1, 2, 3, 4, 5, 6, 7 electrons. In an atom, protons are going to equal electrons. The other thing that we can deduce from looking at all these structures is how many electrons can go in each of those electron shells. The innermost electron shell can hold up to 2 electrons. The next shell can hold 8. The third shell can hold 8. That's as far as we need to go for biology, for the biological chemistry that we're going to talk about that has to do with humans. That's as much as we need to know. The other thing to keep in mind is that atoms are most stable when that outer electron shell is full or complete. Stable means they're not going to undergo any chemical reactions. They're not going to join together with any other atoms. They're not going to make molecules. So in order to figure out what they're going to do, we have to figure out how to make them stable, how to get their outer electron shell to be completely full. Couple of other pieces of information to look at. The atomic number. We saw that in that other figure, and we're going to go back to that figure in just a second. The atomic number defines an element. Anywhere you go in the universe, if you find an atom and you give it the atomic number of 6, it's going to be carbon. And the atomic number is equal to the number of protons in the nucleus. So anywhere you go in the universe, if you find something, find an element that has 6 protons in its nucleus, you can say this is carbon. If you find something and say this is carbon, it better have 6 protons in the nucleus. Now we're going to talk in a minute about the mass number, because that gives us some information about something called isotopes, which are very clinically relevant in human medicine. But I want to stick with this atomic number for just a second. We can put this with what we know about the electron shells, and we can start to draw some of these atoms, and that's going to help us figure out what it is that they're going to do. So the simplest atom that they have here is the hydrogen atom. Hydrogen has an atomic number of 1, so it has 1 proton. Since it's an atom and the number of protons equals the number of electrons, it's also going to have 1 electron. That electron is going to go in this first electron shell. That first electron shell can hold up to 2. So we have an empty space here. That empty space tells us that hydrogen is going to try to fill up that empty space to have a completed outer electron shell, and it will form bonds with other atoms to form molecules in order to do that. So we can do the same thing by looking at oxygen. we can draw oxygen. I want to do that. Oxygen has an atomic number of 8. So it's going to have 8 protons in the center of the atom. And it's an atom, it's going to have 8 electrons also. 2 will go in the first electron shell. The rest will go in the second electron shell. Now they tend to pair up. This is going to show us where some of the empty spaces are, because our outer electron shell can hold up to 8, but there are only 6 in oxygen. So there's an empty space here and an empty space here. Oxygen is going to want to form 2 bonds in order to complete its outer electron shell. And so we're going to come back to that idea in just a minute as we talk about the way that chemical bonds form. The other thing to talk about as we're talking about atoms and the structure of atoms is this mass number. The mass number is going to be the total number of particles in the nucleus, the total number of protons and neutrons in the nucleus. So the mass number, mass is similar to weight. And so what they do is they weigh the nucleus to figure out all the particles that are there. If we have the atomic number and we have the mass number, then we can figure out the number of neutrons. So let's go back and look at oxygen on our big figure. Oxygen has an atomic number of 8, and oxygen comes in 3 versions, the most common of which is oxygen 16. Oxygen 16 is called that because it has 16 particles in its nucleus. 16 protons plus neutrons. But we already know how many of those are protons. The atomic number of oxygen is 8. That tells us there's 8 protons in the nucleus. 16 minus 8 tells us there's also 8 neutrons. 8 and 8 are 16. That gives us our mass number. And so our isotopes, just the way I've written it here, are named based on the mass number. Now for oxygen, that's not very interesting. But for some other elements, that can be relevant. Carbon is an example of an element that has 3 different versions, or 3 different isotopes. Isotopes differ in the number of neutrons in the nucleus. So we have 3 different isotopes of carbon, and we name them carbon-12, carbon-13, and carbon-14. They're named based on their mass number. Now, all of these are carbon, and we've already said anywhere you go in the universe, if you find something that's carbon, it has 6 protons in its nucleus. And if you find something with 6 protons in its nucleus, it's carbon. So all of these have 6 protons. Carbon is an atom, so the number of protons is going to equal the number of electrons. So all of these elements have 6 electrons. What varies is the number of neutrons. And the way we find the number of neutrons is to take the mass number, which is protons and neutrons. It's everything that's in the nucleus, but we know how many protons we have, so the difference is the number of neutrons. 12 minus 6 is 6. 13 minus 6 is 7. 14 minus 6 is 8. This becomes relevant in human medicine because some, not all, but some isotopes are radioactive. That means that some of these neutrons are released from the nucleus in a process called radioactive decay. We can detect radioactivity. And so we can use these as what we call radioactive tracers in the human body. And so there are lots of examples of radioactive tracers that we can use, that we can find where it goes in the body to help us understand how things are functioning inside the human body. And so when you hear of radioactive tracers or radioactive isotopes, those are just versions of the element that vary based on the number of neutrons in their nucleus. So we've talked a little bit about the structure of the atom. And again, we're not really going to talk a lot about atoms individually in human biology. What we are going to talk about are molecules. And so atoms can be joined together to make molecules. They do this by forming chemical bonds, and they do this in chemical reactions. So after we talk about these three types of chemical bonds, then we'll talk about chemical reactions. The first one that we're going to talk about is the ionic bond, and then we'll talk about covalent bond and hydrogen bond. But I've listed them here in a particular order, in the order of decreasing bond strength. So the covalent bond is the strongest of the three. The hydrogen bond is going to be the weakest of the three. Now, so far, I've been pretty particular to talk about atoms are electrically neutral. And I've represented that by saying our number of protons is equal to our number of electrons. And I've said that because there's another entity, ions. Ions are atoms that have either gained or lost an electron. There's a reason why atoms sometimes gain or lose electrons, and it's to become more stable. All of these atoms are trying to undergo chemical reactions to try to be more stable. So we're going to look in just a second at an atom that's going to gain an electron. It creates a negative ion, something called an anion. And we're going to look at an atom that loses its electron to create a positive ion, and that's often called a cation. Little easy ways to remember that. The word anion has an extra N in it for a negative ion. Cation, the T looks like a plus for a positive ion. So let's look at how this happens. So we're going to look at sodium. Sodium has an atomic number of 11, so it'll have 11 protons in its nucleus, and it'll have 11 electrons. The sodium atom is going to do this P equals E. So where do those 11 electrons go? Well, 2 go in this first electron shell, 8 go in the next electron shell, that's a total of 10. The 11th one goes in this outer electron shell, all by itself in that huge outside electron shell. Sodium atom is nowhere close to being stable. If you Google any kind of YouTube videos for elemental sodium and what happens when people expose elemental sodium to wet air or to water, you see lots of heat and lots of light and lots of activity. Elemental sodium, sodium atom, is very reactive. It's very far from being stable. But if it loses one electron, that gets rid of this whole outer electron shell. Its new outer electron shell has 8 electrons in it, and it's much more stable. So sodium has converted from an atom to an ion by losing its electron. It's actually more stable in this ionic charged form than it ever was as an atom. Chlorine does the same thing. Its atomic number is 17. It's trying to have a full outer electron shell. It's trying to be stable, but it ends up having one empty space. Well, if it can take on this electron from sodium, then it can be stable and full and happy. It's taken on an electron, so it has formed an ion. And so we have formed a sodium cation, and we have formed a chloride anion. Now there's a property in our universe that says opposites attract. Sometimes we say that in regards to relationships, but it's definitely true in the laws of physics. And so we have an attraction between these opposite charged ions, and that can form a compound. That can form a molecule of sodium chloride. Now in sodium chloride, as sodium ion, sodium is extremely stable. It's so stable that we put it in salt shakers on our kitchen table where our toddlers could knock it over. As an ion, sodium is not very reactive. As an atom, it's not very stable. So it converts to its more stable form, and that's how we find it in food and in the human body. So that's our first kind of bond, ionic bonds. First we form ions, and then the opposites attract to form ionic bonds. Now sometimes the outer electron shell of an atom has like 4 or 5 or 3 electrons. Too many to give away, too many to take on, so it's hard for that atom to form an ion, but it still wants to be stable. It's still trying to complete that outer electron shell. So instead of fully giving up or taking on an electron, sometimes we can share electrons between 2 atoms to create what we call a covalent bond. And so here's an example of some of those that take place. What we can do is we can look at going back to drawing our atoms. So we can first draw oxygen. Oxygen has an atomic number of 8, so it has 8 protons in its nucleus. We're back to talking about atoms. So 1, 2 go in the first electron shell. 6 more. But that second electron shell can hold up to 8 total, so we have these 2 empty spaces. Think back to hydrogen. Hydrogen has an atomic number of 1, so it has 1 proton and it has 1 electron, giving it 1 empty space. Now here's where I wish I could animate this, because what I would do is I would slide this hydrogen atom much closer, and this hydrogen atom much closer to show you that they could share this pair of electrons. So sometimes both of these electrons go around oxygen, and then sometimes they both go around hydrogen. Sometimes both of these atoms go around oxygen, and sometimes they both go around hydrogen. This forms a compound called H2O, or water. This is how water is formed. This is going to be a single covalent bond because we're sharing a single pair of electrons. There's a single set of shared electrons between each of these atoms. These are going to be examples of polar covalent bonds. So here's where I introduce a new term, and that term is electronegative. Some atoms are very electronegative. That means they hold on to electrons very tightly. So now my drawing is going to get really messy. So instead of just one trip around each of these atoms, these electrons will spend a whole lot of time around oxygen for every time they go around hydrogen. A whole lot of time around oxygen for one trip around hydrogen. And so all these black lines are telling us that the electrons tend to be around oxygen a little more frequently. Not enough to make an oxygen ion, so it's not like a full negative charge. It's what we call a partial charge, and we denote it by using a lowercase Greek letter delta. And that makes the electrons not be around the hydrogens as often, and they get a little bit of a positive charge. The word polar is going to mean difference across two ends. Different. All right. That's what I get for trying to talk and write at the same time. They're different on one end than the other. We're going to use that word polar again when we talk about the cell membrane. The cell membrane is different on one side than the other. And so this polar covalent bond is different at one end than the other. It's a little more negative at one end and a little more positive at the other. And that happens when we have electronegative elements involved in the covalent bond. So in this image of water at the bottom, they're showing the electrons stretched toward the oxygen molecule. Rather than being equally shared, carbon is not electronegative, hydrogen is not electronegative. They play well together. They share their electrons evenly. Oxygen, not taught to share yet. And so elements that are electronegative usually form these polar covalent bonds. The polar covalent bonds are bonds that are different at one end than the other. And this sets up the conditions for our third type of bond, hydrogen bond. So those polar covalent bonds occur when hydrogen covalently bonds with elements that are electronegative, and it sets up on one end a little bit of a positive charge, and on the other end a little bit of a negative charge. Remember our rule of physics and sometimes relationships? Opposites attract. So we're going to have an interaction, an attraction between these two oppositely charged parts of different molecules. So here's our water molecule. And since oxygen is electronegative, it takes on a little bit more of the electrons some of the time. takes on this partial negative charge. And so all of these oxygens have this partial negative charge. And that leaves these hydrogens to have a little bit of a partial positive charge. And so we can get this opposites attract interaction, this dotted line that's showing us this interaction between the partially negatively charged part of the water molecule and the little bit of a positive charge on an adjacent water molecule. Places where we find hydrogen bonds are going to be in between water molecules. That's what they're showing you here. And anything else that has these polar covalent bonds, like glucose, proteins and amino acids, nucleic acids. These are all examples of polar molecules. So on the previous slide, I have written hydrogen bonds form between molecules or parts of molecules and can help stabilize structures. We're going to see that in the double helix that is the DNA, the bases are held together, the two strands are held together by hydrogen bonds. We're going to see that proteins have to be folded properly in order to function, and that folding is stabilized by hydrogen bonds. So here's a slide that gives you a summary of the three types of bonds that we've talked about. We started by talking about ionic bonds. Ionic bonds form between ions. So first we had to talk about how we create ions. We lose or gain an electron. And then the actual bond is formed because of this opposites attract principle. If we can't form an ion, our next option is to share electrons to complete the outer electron shell of both atoms. Since they're sharing electrons, this is the strongest of our bonds. We can have nonpolar covalent bonds. Those are just regular covalent bonds. That's just equal sharing of electrons. Or we can have polar covalent bonds, which is unequal sharing of electrons between the two atoms forming the covalent bond. And then in those molecules that contain polar covalent bonds, we can form these hydrogen bonds. All of the molecules that we will talk about in the rest of this chapter and all of our chapters to come will form by one of these three methods. They will either form by covalent bonds, polar or nonpolar, hydrogen bonds, or ionic bonds. Most of the time, the molecules that we'll talk about are either ionic or covalent, and then the structure of the larger molecules is usually stabilized by the hydrogen bonds. So now we can move on to some what we tend to call inorganic chemistry. Your book has a whole page, and we're going to talk in just a second about how water is often a good solvent. Water aqueous solutions are easy to form. And so we're going to build on what we've been talking about how water forms hydrogen bonds and how water can be used to dissolve a number of substances. And so we need to talk a little bit about terminology related to solutions. In the human body, water is an extremely important constituent. And so a number of our solutions in the body are what we call aqueous solutions. They use water as the solvent. So a lot of times when I teach terminology related to solutions, I'll ask students in my face-to-face class, I'll say, how do you make Kool-Aid? Somebody tell me how you make Kool-Aid. And usually somebody comes up with, well, you open the packet and you dump it in the pitcher, you add some sugar, and you add some water. And so Kool-Aid is a solution. It's going to be a mixture of two or more substances. It's an aqueous solution because water is what we're adding. And so the two parts that we're mixing together to make a solution are the solute and the solvent. And so in our Kool-Aid example, the color and flavor molecules from the packet and the sugar molecules that you add, those are all the solutes. Then you dissolve it in the water. The solvent is what does the dissolving. The solute is what's being dissolved. Now there's a number of organic solvents. If you've ever bought the product Goo Gone to try to get sticky stuff off, Goo Gone is an organic solvent and has a lot of petroleum distillates so that it does not have a lot of polar covalent bonds. And so it can try to dissolve adhesives and things that are really sticky. Saline solution is an aqueous solution. Sodium chloride is the solute in saline solution. So this gives us some terminology to work with as we talk about these things that are dissolving in water to make these aqueous solutions. So in a solution, the solvent dissolves the solute. If substances contain polar covalent bonds, they're going to dissolve very readily in water, and we call them hydrophilic, liking water. Substances that contain non-polar covalent bonds are going to be called hydrophobic. This is going to be things like lipids, the phospholipids that help form cell membranes, steroid hormones, cholesterol molecules, triglycerides. Water has the polarity, can form the hydrogen bonds, and so it can interact with lots of ions or molecules. That's one of the things that makes it a very good solvent. So here's our water molecule, and remember, a little bit of it is partly negative and a little side of it is partly positive. And so when we put some sort of ionic compound in there, we can get what's called a hydration sphere, which is just what happens when we which is just what happens when we mix. Did I do that right this time? There we go. When we mix an ionic compound with water. And so what ends up happening that over here. What happens to us when we form this hydration sphere is that the partially negative parts of water surround the positive ion. The partially positive aspects of the water surround the negative ion. And that helps to pull these ions apart. Here's how that helps. When you're trying to boil water, you tend to add salt to your water. That breaks up the hydrogen bonds in the water and allows it to boil a little bit faster at a little bit lower temperature. This is also the way that salt melts ice. Ice is water whose temperature has been lowered to the point where those hydrogen bonds tend to become more solid. And so if we can break those hydrogen bonds, then we can melt the ice. And so we can add sodium chloride, other salts like potassium chloride, magnesium chloride, calcium carbonate, all of those form ions when they're dissolved in water. And those ions can help pull that water out, break those hydrogen bonds, and melt the ice. The other thing that we talk about when we talk about solutions is their pH. pH stands for potential hydrogen, and so this is the potential for how much hydrogen a solution will release. When we talk about pH, all solutions fall into one of three categories. All solutions are either neutral or acidic or basic. And they're put into this category based on their concentration of hydrogen ions. So if something has a pH of 7, which actually means the hydrogen ions are equal to the hydroxide ions, then we say that solution is neutral. Anything that gives off more hydrogen ion has a lower pH, and so we say that's very acidic. Anything that gives off a lot of hydroxide ion has a higher pH, and we say that that's very basic. So let's take our time and go through this and kind of unpack some of those details. The first thing to look at is, what is this hydrogen and hydroxide ion when we put it together? A hydrogen ion plus a hydroxide ion forms a water molecule. Water is completely neutral. Pure water is the most neutral substance. So if we have a substance, or if we can make a substance where the hydrogen ion and the hydroxide ion concentration are equal, those will combine to form water. If we have more of the hydrogen ion released in solution, that's giving us an acid. And if we have more of the hydroxide ion released in solution, that's giving us a base. The pH scale runs from 0 to 14. And so that's how 7 gets to be this neutral pH. Anything between 0 and 6.999999999 is acidic. And anything from 7.1 to 14 is a base, or is very basic. And let me give you a couple of other terms. Basic, another term for that is alkaline. So you can talk about having things that are acidic or alkaline. And we can talk about the acidity or the alkalinity of a substance. Now your book gives you a really good figure. It actually orients it up and down. And it gives you the pH of a number of different substances along that pH scale. So a lot of the things that are going to be more acidic are going to have a lower pH. Stomach acid has an extremely low pH of around 1. Battery acid, sulfuric acid, and drain cleaner, which is also sulfuric acid, is going to have a very low pH. On the other end, things that are going to be very basic or have a very high pH are going to be things like baking soda, sodium bicarbonate, and then things like bleach and oven cleaner and those types of really caustic cleaning solutions are going to be ones that are going to be very basic, that are going to have a lot of hypochlorite and those types of substances that can tend to be very basic. Most of the substances in the human body kind of stick to this 7, or slightly more acidic or basic. Normal blood pH is about 7.2 to 7.4, so that's pretty close to neutral. And most of our body fluids are going to have more of a neutral pH. The biggest exception, as I just mentioned, is stomach acid, which is going to have a very low pH. And then in the digestive system, we're going to have a way to neutralize that really low pH after it does its job. So to give you some definitions, to put some of what I was just talking about into words, acids release hydrogen ions into solution. The definition of bases is that they remove hydrogen ions from solution. They do that because they release those hydroxide ions, which combine with the hydrogen to form water. Now, one that we're going to look at is carbonic acid. And so in your book, at the bottom of page 29, it gives you this thing that we're going to study that's called the big equation. When carbon dioxide dissolves in water, it forms carbonic acid. And that very readily dissociates into a hydrogen ion and a bicarbonate ion. There's lots of places in our body where carbon dioxide is produced. So there's going to be lots of places in our body where this physiological mechanism is going to take place. It can be sped up by an enzyme, and that enzyme is called carbonic anhydrase. We're going to talk more about enzymes in just a second. This is how your respiratory system maintains acid-base balance of the blood. This is one way that the urinary system contributes to acid-base balance of the blood. This is going to be a major homeostatic mechanism that we're going to talk about. We're going to go through this a couple of times this semester. So if you just went, oh my God, what did she just say? Don't worry, I'll say it again several times. But that's usually what I refer to as the big equation, because we can use it kind of as a theme as we go through our semester and talk about acid base in each of these systems. So to give you an image of what we were just talking about, acids release hydrogen in solution. So hydrochloric acid, HCl, KCl, all of those. No, sorry, that's not the one. That's a salt. Our carbonic acid, H2CO3, that releases hydrogen ion. All of those that release hydrogen ion are considered to be an acid. Sodium hydroxide, potassium hydroxide, magnesium hydroxide, anything that's going to release hydroxide into solution is going to be a base. And again, the technical definition is a base removes hydrogen ion from solution by releasing that hydroxide, because when you mix hydroxide and the hydrogen ion, you get water. Let me actually write this so you can see what it is that I'm trying to say. Hydrogen ion plus hydroxide ion yields a water molecule. And then this is a chart of the pH scale. I like the one that's in your book, which gives you the same information, but it also adds in some of the compounds and where they fall, like battery acid is very acidic, and oven cleaner and drain cleaner are very alkaline. I like this because it shows us neutral. It shows us increasingly acidic and then increasingly basic, and it also gives us that alkaline term. This also helps me kind of as a visual person understand that the farther down this acidic part we get, the more hydrogen ion is in solution. And the further we get on this basic, the less hydrogen ion we have because that hydroxide has taken it out of solution. It can be a little counterintuitive as the numbers get smaller on the acidic side of the scale for us to say more acidic. So 2 is more acidic than 3, even though the number is lower. 1 is much more acidic than 4, even though the number is lower. The numbers make more sense on the more basic side. So take a look at the one in your book and see if you can make that make a little bit of sense in your head. And then here's another one that's nice and blurry, but it has lots and lots of examples of different substances on the pH scale. And again, this is showing alkaline is more toward pH 4, and acidic has the lower pH. And so some of these pH amounts can be altered a little bit in the human body because we have buffers that can remove or replace the hydrogen ions in solution. And so those buffer systems help maintain the pH of body fluids. And as we go through each system, that's where we're going to talk about the big equation, how it's going to work as buffer systems to help try to maintain homeostasis of pH of body fluids. So to conclude our discussion about pH, we can look at this question. What do you get if you add equal amounts of sodium hydroxide, which is a base, to hydrochloric acid, which is an acid? Well, in solution, both of these compounds are going to ionize. So in solution, you actually have a sodium ion and a hydroxide ion, and a hydrogen ion and a chloride ion. And so if you titrate exactly equal amounts, the hydrogen ion and hydroxide ion will form salt, or will form water. I just gave away the answer. And the sodium and the chloride will form table salt, sodium chloride. So if you add equal amounts of sodium hydroxide and hydrochloric acid, it won't explode, it won't get hot, won't turn colors. It's just going to turn into salt water. So this is the last part of the chapter, and it starts to go through a whole set of the organic compounds that are important to human biology. And I'm actually going to record this as a separate lecture because I know that this is a lecture that people are going to want to kind of come back to as they work on the discussion board and the assignments, both this week and in future weeks, as we start to talk about things like the digestive system and cell function and those kinds of things. So the rest of the chapter is going to talk about these 4 major classes of molecules. This is what we've been working up to this point in the chapter. So we started to talk about atoms because we needed to talk about how atoms get put together. The central focus of that discussion was the bonds and how bonds form. Because as we make these big macromolecules, we're going to be forming and breaking covalent bonds. And as we fold these molecules or we dissolve them in water, we're going to need to talk about hydrogen bonds as well. So we had to talk about the structure of the atom to get us up to the bonds and how bonds form. Then we had to talk a little bit about some basic chemistry terminology, about solutions, solute and solvent and aqueous solution, hydrophobic, hydrophilic, pH, acid base, some of that basic terminology. And so all of that has brought us to this discussion of these 4 major types of organic compounds. Now, when we define life, a lot of textbooks include in their list of characteristics of life things that are comprised of and make or synthesize these organic compounds. So these are going to be really, really key to the rest of the semester and our discussions in a number of the systems. So if you have any questions so far on Chapter 2 material, or at the end of the next lecture, if you have questions about the remaining Chapter 2 material, please be sure to post on the discussion board or send an email to the whole class or to me. There's lots of us that will be able to hopefully get you an answer to your questions.